Hey everyone, my name is Duncan Davis Hall. I'm a PhD student in the Magan Lab for Bioinspired Pulmonary Engineering at the CU Denver Anschutz Medical Campus Department of Bioengineering. And my project, my research, is based around 3D bioprinting models of the blood vessels that are in your lungs that I can use to study diseases that happen in blood vessels in your lungs. So I'm going to walk you through the process of 3D bioprinting. Today is going to be mostly weighing things out and getting everything ready so that we can actually do the printing tomorrow. And that will involve some cells, it will involve some polymer chemicals, and in the end we're going to have a final product that is a soft hydrogel, which is a polymer biomaterial, with lung cells inside of it that I can then use to study diseases of the lung. So we're going to go into the lab, we're going to get all the stuff out that we need to weigh, and we're going to do a bunch of prep work today so that tomorrow we can do the actual 3D bioprinting. And if everything goes okay, we're going to end up with a final product that I can use for my research. So we're going to head on in there and get everything going right now, and hopefully it all works out. So the first thing that we need to do before we start 3D bioprinting is weigh out all of our materials. You know, chefs do this, they get everything for their recipe out before they make it so that they know that they have everything that they need to make sure that it works. So here I'm at the balance and I have a lot of different materials that I'm going to be using to make these three-dimensional hydrogels to put cells in. The first thing that I'm going to weigh out is called life support. This is a gelatin microparticle powder that I'm going to weigh out, and then tomorrow I'm going to dissolve it in cell culture media to make a support bath for the 3D printing. This will all become very clear tomorrow. Today, all I need to do is weigh it out, and all I need is 1 gram. So I'm going to grab a weigh boat for the balance here, and I'm going to weigh myself out 1 gram of life support. Balances are pretty precise, this one is at least. We always make sure that it's leveled before we use it. If it's out of level, then it can give you an incorrect reading. So make sure that it's leveled, everything's green, and then we can go from there. So switch it over to grams, and we'll zero it with the weigh boat on there. This says that my weight is now 0, so it's not counting the weight of the weigh boat. And this stuff is pretty easy to weigh out. I need a lot of it, so I can just kind of dump it in this way until I get up to 1 gram. And this doesn't need to be exactly precise because I'm going to be dissolving it in a fair amount of media, and I just need enough of this to print into tomorrow. So this is more than I need really, but it's good to give yourself a little bit of wiggle room. We'll just wait for that to equilibrate there. There we go. About 1 gram. So we'll take this out and I'll put it in a fresh empty conical. And then label it. Labeling everything. So I'm going to be leaving this overnight. There's all sorts of other people that are doing all sorts of great research here, and I want them to know that I made this. I want them to know what it is and when I did it. So I'm just writing on the side here what this is. It's 1 gram of life support. It's got my initials on it, and it's going to have the date. All right. That's all we need for that. So we'll put that away and keep our space pretty clean here. And we'll weigh out all the materials for the Bio-Ink now. So what I just weighed out was life support, which is the slurry that we print inside of. What we're actually printing is going to be a Bio-Ink solution, which is going to be a hydrogel that we then put cells in. So the Hydrogel Bio-Ink has a few different components. This is PEG alpha methacrylate. It's a chemical that we make in the lab that it forms the backbone of our 3D bioprinted structure. So the majority of what I'm printing is going to be this PEG alpha methacrylate. So that makes a backbone. It's an 8-armed kind of star-shaped polymer, and we need to find a way to link those arms together to make it actually solidify so that the cells can live in it. So we have a couple of different crosslinkers here. This one is fully synthetic. It's called dithiothreatol. We buy it from a company, and it's very short. So it's going to link the arms of this PEG alpha methacrylate together. We're also going to use a little bit of this Mmp Degradable peptide crosslinker. Now, that's a lot going on in that phrase there, but basically, this is a chemical that cells can degrade with something that they secrete called matrix metalloproteinases, or MMPs. So we call it an Mmp Degradable Processor. And it's a chain of amino acids, just like you would find in proteins in your body, that can link our PEG alpha methacrylate and then allow the cells to dissolve it and remodel their matrix. So we'll weigh all those out too, and we're going to use much smaller tubes for this than the big weigh boats that I use for life support, because instead of needing a gram of all of this stuff, I need milligrams of all of this stuff. So I'm going to label my tube again and pop that on balance there. Switch everything over. Zero balance. So the PEG alpha methacrylate is going to make up the bulk of this bio-ink, so I'm going to be weighing out more of this than anything else. And PEG stands for polyethylene glycol. It's a fairly common synthetic polymer. It's bioinert, which means that your body doesn't really register it, which is why we put in some other things to help the cells do their thing once they're inside of the polymer. And I wrote down in my lab notebook the amounts that I need for all this, so if you see me looking off to the side, I'm checking the numbers that I wrote down. These recipes are very, very carefully formulated and validated. We do a lot of other experiments in our lab to know exactly how much of everything that we need to make this work. Because again, we're trying to create a 3D bioprinted structure that models the lung blood vessels so that I can research hypertension, which is high blood pressure in the lung blood vessels. So I know how much of this I need, and I'm just going to be kind of adding and checking. Pretty close there. Basically everything I'm weighing out today looks like the same kind of white powder, but at the chemical level, there's some very key differences that allows it all to work. And I need just a little bit more of this PEG alpha methacrylate. Okay. So there's a certain minimum amount of this that I needed, and everything that we do in the lab, you don't want just the minimum, so I weighed out a little bit more than I am going to need tomorrow. And I'm going to label the tube with the actual mass that I got, as well as writing it in my lab notebook, so that tomorrow, when I go to do all the printing, I know exactly how much I have. And before we weigh out the next chemical, I'm going to clean up my workspace here a little bit to make sure that none of the PEG alpha methacrylate that I just weighed out gets into the tubes of the other chemicals. So, for that, we use a little bit of acetone. This is in nail polish remover, and it's going to dissolve any extra PEG alpha methacrylate that I have here. It dries very, very quickly so that I can then use this to weigh everything else out. And we're going to repeat what I just did with these other few chemicals here. I need much less of all of this stuff. What I'm doing now is weighing out the crosslinkers. So I just weighed out the polymer backbone that makes up the majority of my bio-ink. And now I'm going to weigh out the chemical crosslinkers that are going to link that backbone together to create a solid hydrogen. Keep everything wrapped up because we don't want it to get any water from the air inside of it. That gives us all sorts of problems. So what I'm weighing out right now is called dithiothreatol. That's the chemical name. We call it DTT for short. And it is a very, very short crosslinker. By that I mean that the actual chemical arm is very short, which helps us makes our Hydrogel Bio-Inks, makes it more solid. So I just need a little bit of this. This is probably plenty already. Yeah, that's plenty. Again, I'm going to write down the actual mass that I weighed out here so that tomorrow I know exactly what to do with it. And we'll wrap everything back up. Clean off our spatula here and move on to the next chemical. So the next chemical is the Mmp Degradable Processor that I mentioned, which can be degraded by cells to allow them to make their environment more comfortable. We don't use entirely degradable Crosslinker because then potentially the whole 3D bioprinted structure could fall apart if the cells degrade everything. So that's what the DTT was for that I just weighed out. That is going to stick around so that the cells don't get rid of everything. And this is a larger chemical, a longer chemical than the DTT. So I'm actually going to be using more of it. Seems a little counterintuitive, but longer chemical means that any given mass of this chemical has fewer individual crosslinkers inside of it. So to get more crosslinkers, I need more mass. Oops. We'll see how much we got there. And that's enough. Recording everything as we go. I put in my recipe in the lab notebook before I started this, so I knew exactly how much of everything I needed, and I'm writing it all down as I go, just to make sure I don't lose track of anything. And this is all preparatory work. Essentially trying to get as much ready ahead of time as I can before I start the actual printing process. The last thing that we're going to weigh out is another peptide, so it's a chain of amino acids, and which is what your proteins are made up of in your body. And this peptide we call RGD. Those are the abbreviations of the amino acids that make it up. And that group mimics a protein found in your body called fibronectin, which cells like to attach to. So most of what I've weighed out so far, cells can't attach to, but I want to put cells into my 3D bioprinted hydrogels. So I'm putting in a little bit of this fibronectin mimic, and the cells will be able to bind to that through proteins that they express called integrins. This is how cells attach to things in your body. So we'll weigh out just a little bit of this RGD. This doesn't help hold the hydrogel together, but it helps the cells attach to it. This is a very, very small chemical, very short, which means I only need a little tiny bit to have a lot of individual chemical groups. That's probably too much. We'll see. All right. And record everything. Wrap it all back up. This is basically fancy lab saran wrap that prevents air and water from getting into these tubes when we store them in the freezer. And that is everything that we need weighed out. So we weighed out some gelatin microparticles called life support, which is going to be what I print inside of to hold everything together. And then we weighed out 3 different hydrogel components, a polymer backbone, and 2 different crosslinkers, and we weighed out a small adhesive peptide that the cells can use to attach to. So these will all be stored in the freezer, wrapped up to protect them from air and water, and then tomorrow I'll be dissolving everything and using it to make the bio-ink that I'm going to put cells in to 3D print. So we just weighed out all the chemical components that I'm going to need to do 3D bioprinting, and now I'm going to prepare the solvents for those chemical components. So we just had a bunch of powders and solids, but I need those to be liquid to make a bio-ink to put cells in to 3D bioprint. So we're going to get a few different kinds of cell culture media ready. This is a bottle of cell culture media. It's basically cell food. It's what we feed cells to keep them alive. And I'm going to use this in a couple of different ways to 3D bioprint my hydrogels tomorrow. The material that I am 3D bioprinting, the PEG alpha methacrylate that I keep talking about, it will polymerize in basic conditions. And by that I mean a basic pH over about 8 or so. So I'm going to make 2 different solutions of cell culture media, one that is at pH 9 and one that is at pH 6.2. So the pH 9 media is what I'm going to dissolve my gelatin microparticles in to make a support bath to print inside. That high pH, that basic pH, will allow the bio-ink to polymerize with the cells inside. The bio-ink itself I need to be at a low pH, because if it's too high, then it will polymerize on its own before I get it where I need it to be. So that's what the pH 6.2 media is going to be. That's slightly acidic, and so that will keep the bio-ink liquid until I put it into the slightly basic support. This will all become really clear later. Today I'm just getting everything ready. So I'm going to take some of this cell culture media and I'm going to measure it out so that I can pH it. I'll try to do all this over here. I got a couple of tubes here I'm going to be using. So you may have noticed that we're in a different location. This is a biological safety cabinet. This is where we do cell culture. It's a sterile environment. And there's something called sterile technique that's very, very important in here. But basically, whenever you're working in here, you want to keep everything really, really clean. And this will help keep our cells alive, our cells alive as well. So, this media is not normally the pH that I need it to be. I need it to be pH 9. I just measured some out, so we're going to see what pH it's at. Once we figure out this is actually touching. Alright, we're good. This is the pH meter. It'll tell me what the pH is. And I'm going to go grab a couple of things to help me get a couple of other solutions in here that will get us to the right pH. It's hard to see right now, but I'm spraying things down with 70% ethanol before I put them in here. It's a decontaminant. So our media, after we dispensed it, is a little low. This is a pH of 7.8 and I need it to be 9. So I'm going to rinse off the probe here. And then I'm going to add a little bit of a slightly more basic solution. So basic means higher pH. I can take a little bit of this. I can put a little bit of that into here. and see what we got. So what these are going to help me do tomorrow once I get them to the right pH is to make sure that my bio-ink stays liquid when I need it to and then becomes solid. And what we're going to use these to do is dissolve all of those powders that I had before, like I mentioned. So having these at the correct pH is pretty crucial, because if they're off by a little bit, then I can end up with having a solid where I'd rather have a liquid, or having a liquid where I'd rather have a solid. So making sure that this is all very precisely done is pretty important. We won't go through the whole procedure for pHing this, because it takes a little while. We see that we went from 7.8 to 7.9, and I need 9, so I'm going to need to do this a few more times. But I'll quickly show what I'm going to be doing with the other batch of media. You can actually see the differences here. So this is the normal cell culture media. It's orangish, and it has an indicator in it that will change color in different pHs. So we have our high pH basic media that's becoming a little bit more pink, a little bit more purple, and our low pH acidic media, which is more yellow. Normally this is a sign that the media needs to be changed and that the cells are having a hard time, but there's no cells in here, so I'm not too worried about that. But this is going to be the slightly acidic or low pH media that I use to make the bio-ink. So we'll just very quickly check on that. And I think I need to do it this way. I want this to be about 6.2. And I made it in advance, so it should be close. Looks like this one's going to be a little bit off as well, but there's two solutions that I have here, one with a high pH and one with a low pH. They're very cell-friendly, that I can use to get everything where I need it to be. So we'll get everything where we need it to be for tomorrow. We're not quite there yet, but it's just a matter of adding a little bit more and then measuring again. And then now we're going to have everything ready that we need for bioprinting tomorrow. So we'll have our dry chemical components that are going to make the hydrogel. We'll have our liquid media that is going to help us dissolve all that dry stuff, and then we'll show you the cells tomorrow that we're going to put inside. We'll set up the bioprinter in the biological safety cabinet, and we'll get printing. All right, Duncan Davis Hall. We are back here in the Magan Lab for Bio-Inspired Pulmonary Engineering at the Department of Bioengineering at CU Anschutz, and we're going to pick back up where we left off, 3D printing. So I showed you before some of the prep work that goes into 3D printing, where we weigh out a series of materials that is going to be used to make our 3D printed hydrogels. And today we're going to put all that stuff together and actually do the 3D printing. So we'll get all of our chemical components dissolved, we'll get them combined, we'll prepare a support bath to 3D print inside of, and then we'll passage cells to put in the bio-ink and then do the printing. So the first thing we're going to do is prepare a gelatin microparticle support bath. So yesterday I weighed out a gram of life support. Right now it's just a dry powder, but this is these are gelatin microparticles. Think of Jello. So gelatin is going to be the support material that will hold our 3D printed hydrogels while the printing is happening. So I'm going to prepare that today by dissolving it in a small amount of the cell culture media that I made at pH 9 yesterday. So again, this is slightly basic. The pH is above 7. And that is going to help our 3D prints polymerize or solidify after we deposit them. So we're going to get that going here. And pretty much everything we're going to do today, whenever tubes are open, we're going to try to keep them open here in the biological safety cabinet to help keep things sterile and make sure that our prints actually can support cells. Some media and add it to our life support. So this needs a lot of mixing and stirring and soaking to get up to a point where we can actually use it. Right now you can see that all of the gelatin is kind of down at the bottom and all the media is kind of up at the top. So we're going to vortex this to get it all combined, and then we're going to let it hydrate and spin it down. All right, so the life support gelatin matrix is now vortexed and it's sitting in the refrigerator. It's got to hydrate for about 10 minutes. And while we wait for that to do its thing, we're going to dissolve all of our polymer components. So again, yesterday we weighed out PEG alpha methacrylate, which is a polymer backbone. It's going to make up the majority of the hydrogel. We have two different kinds of crosslinkers, a synthetic crosslinker and a Mmp degradable crosslinker that the cells can remodel when they're inside of the bioprint. And lastly, we have our RGD peptide sequence, which is just a little adhesive peptide sequence that cells can attach to. And I did some math yesterday to figure out exactly how much of all of this I need. Think of it kind of like a recipe. We need a certain amount of all of this to make sure that the hydrogel actually comes out. And so I did some of that yesterday just to know how much of it I need. So we're gonna just dissolve everything here with different pipettes. So these are all dry powders right now, and I need them to be in liquid solution before I can combine them all to do the 3D printing. So I have here my cell culture media at pH 6.2 that I made yesterday, and this is slightly acidic, slightly below 7, and this will prevent my polymer from polymerizing or solidifying before I do the printing. So this will keep everything liquid, and the cells will be okay in here, and then we'll deposit everything into the life support and it'll solidify. So we just take out a careful amount of this and we add it to our polymer backbone here. It's, again, you can see, you may not be able to see, but it's not mixing in super well. So once I get liquid added to all of these powders, I'm going to take this out and I'm going to vortex it or spin it, and I'm going to sonicate it in some warm water to help get all of this solid material dissolved in the liquid material. We don't want chunks of this stuff floating around or it won't work super well. There is another solvent that I'm about to use for the crosslinkers. And this solvent is not cell culture media. Instead, it is it's largely saline, phosphate buffered saline, or PBS, which is basically salt water, and it's got some of a chemical called T-set in it that's going to help dissolve these small crosslinkers. So we'll use that for our crosslinkers. And again, there's a very carefully formulated amount of liquid that goes into each of these tubes of solids. If you are off on this part, then sometimes the printing doesn't work out. So we'll carefully add these solvents to our material. And we'll let these all do their thing. So basically, these liquid solvents are just going to dissolve the powder so that eventually we'll have just a liquid. And once they're all dissolved, I will be taking all of these different components and I'll be mixing them together again in a fairly strictly calculated way to get the Bio-Ink solution. So there's a lot of math that goes into it, lots of spreadsheets. And in the end, we'll have a solution called Bio-Ink that will have all of our polymer components and our cells. And we'll use a little bit more of this media. It's a very small amount of liquid in this last one, but get everything where it needs to be. Okay. So that's all of the solvents added to all of our polymer components. Now I'm going to take it out, I'm going to spin it up, I'm going to mix it down, I'm going to sonicate it, I'm going to heat it, and then it'll all be dissolved so we can use it for our Okay, so we've got our life support slurry all hydrated. Now everything's all mixed together, and this is going to be the support material that we do our 3D bioprinting into. We've also got all of our polymer components here fully dissolved, and we're going to mix these together to make the Bio-Ink Precursor. So, I'll show you how this life support works. We'll do the actual printing inside of a 12-well plate, which is fairly standard piece of plastic for cell culture science, and I'm going to fill some of these wells with the life support. So to get all that out requires a very complicated set of tools. I have a unbent paper clip, a syringe, plunger, and a razor blade. So we're gonna just slide the plunger with the unbent paper clip into here. The paper clip is just to let air escape while I push this down, because I want this plunger to go all the way down to the top of the support. There we go. Take our paper clip out if I can grab it. Otherwise we'll leave it. It's not too big of a deal. Yeah, we'll leave it. I'm gonna take the razor blade and remove the end of the plastic tube here to make a little life support syringe of our own. The reason that takes all this sort of strange mechanical work is just to avoid introducing any air bubbles into this, because air bubbles are very hard to get out of this, and if the printing hits an air bubble, then the shape of my construct won't be right. But we're just going to take this and squeeze out a little bit of life support into some of these. It's not quite as large as I would like it to be. Shave that down a little bit. There we go. It's better. All right. There we go. It's easier if I just do it this way. So I'm planning to make 8 prints today. That's how many I need for an experiment that I'm running. We're going to be taking RNA out of these to see what kind of genes they're expressing. So I'll fill up 8 of these wells. Pardon my arm for a moment here. This takes a little bit of muscle. It's coming out of the coming out where the paper clip is. Let's back it up a little bit. Come on. Nuts. Oh well, that's okay. We have enough. Not everything works out the way that you want it here. Sometimes you just have to scrape the life support off of your syringe. That should be okay, honestly. So, another very complicated science technique to get this down to the bottom. And that'll do it. So, not the prettiest, but we got all our life support in there that we need. And we can set that aside for now. I'm going to mix up these polymer components now. And again, I wrote down the recipes yesterday after I weighed them out, so I just need to combine everything together. So we'll grab, this is our PEG alpha methacrylate. This is the backbone. So we need a lot of this. And we'll put this in a tube, and to that we are going to add another kind of polymer called polyethylene oxide, or PEO. This is extremely viscous and very thick. You can't really tell, but this is a liquid. It's called shear thinning. It's like a toothpaste or ketchup. So it acts like a solid until you apply a force to it, and then it acts like a liquid. And this chemical is going to help us out by making our Bio-Ink shear thinning. And what that does is it makes it so that when it's in the tube, the cells don't all settle to the bottom like they would in pure liquid. The cells will float during the printing process, but since it's shear thinning, when the printer applies a force to it, it'll come out of the needle. And we'll see all that in a little bit, but just for now, I'm going to combine it with my PEG alpha methacrylate. I'm using a positive displacement pipette for this. These other pipettes aren't quite powerful enough to bring up this really, really goopy liquid. So we'll add that in there, and we'll mix it by just pipetting up and down. I can see it floating around in there, so I'm just mixing it up this way to get it all distributed. Okay. And then in our second tube here, we're going to combine our crosslinkers and our adhesive peptide. The reason we're going to keep these separate is that the PEG alpha methacrylate will start reacting with the crosslinkers when they meet. And it's a very slow reaction, but it'll happen anyway. And if I'm not careful, my bio-ink can solidify before I get it through the printer. And I've taken some steps like using acidic media to avoid that, but just to be absolutely safe, I'm going to wait until I'm immediately ready to start printing before I mix the PEG alpha methacrylate and the crosslinkers. But I can mix all the crosslinkers together, so I'll do that. And I'm just glancing down at this recipe here next to me. And again, these all of these chemicals were dissolved and then stirred up, sonicated, spun down. We just have a lot of different ways to mix them. Make sure that everything we need gets in there. All right. And lastly, we'll grab our adhesive peptide. Just a little bit. Okay, so all my components now are in 2 different tubes. I'll basically just mix these 2 tubes together when I'm ready to start printing, and we'll add some cells to them. So we're gonna get the cells out, we're gonna get the printer out, and we're gonna passage those, get the cells up and away from where they're growing right now so that I can put them in my bio-ink. And then we'll get started on the real fun part. So here's what the cells look like when they're growing normally in culture. This is a culture flask. It's made out of plastic. It's very, very stiff. The cells we can see on this screen here, and I can kind of pan around and show you. These are human pulmonary artery adventitial fibroblasts. All that is to say, these are cells from a person's lung blood vessels. So they're growing in here. They've reached the point where they need to be passaged or split, and I'm going to use them to print today. So I'm going to rinse them out and then put a chemical on them called trypsin that's going to make them all detach from the plastic that they're growing on, and they're going to ball up and start to float. So I'm going to take all the cells from off of this plastic culture plate and then put them into my bio-ink. And these cells will go from being very spread out on this plastic to being embedded in a hydrogel for my experiments. Yay, back in the hood. Biological safety cabinet. We got our 2 flasks of cells. There's some culture media in here, and then all the cells are on the bottom surface. I'm going to aspirate or suck out the old culture media, and then I'm going to rinse the cells, and then I'm going to trypsinize them, and that's using a chemical called trypsin to get them up and off of this plastic so that I can use them for 3D printing. So we just take all this old media out with a vacuum tube here, and then we're going to grab our pipetter. I'm going to rinse these cells with sterile phosphate buffered saline, and rinsing them is going to remove any residual media, and that's important because cell culture media, that orange tube right there, has proteins in it that can deactivate trypsin. And we need the trypsin to work on these cells to lift them up. So we'll just do a quick little Pbs rinse here. Put down our header and take the Pbs out. This is also helping to remove any dead cells that might be floating around. I only want live cells, and you can't see them, but they're on there. Now we'll go into our trypsin. This is a chemical that will separate the cells from the plastic. Cells are not happy being separated from the thing that they're growing on. To the cells, that is a symbol that they might be dead, or maybe they should die. So they're not super pleased about it. So we'll only leave this on for just as long as it takes to get them up and off the plastic. And then we'll take them and we'll put them in the Bio-Ink. Alrighty. So that's going to go in the incubator, and in a little while we'll take all the cells out, count them, and throw them in the Bio-Ink for printing. You can see that the cells have been trypsinized. Before they were all spread out, they were all attached. Now we can see they are quite round. They're still in their flask here, and if I jiggle this around a bit, we might be able to see them floating somewhat. Yeah, there we go. So if I zip around, I can see that these cells are round and floating. So we'll take these cells out, we'll count them up, and then we'll put them in the Bio-Ink for 3D printing. Got our cells, they're floating in trypsin. Now we need to deactivate the trypsin and get the cells out of here. Because again, it's not super great for them to be sitting around for too long. So it's only been 7 minutes or so since I put the trypsin on, and I'm going to rinse these flasks, wash out the cells with cell culture media. So I'll just grab some cell culture media here, and we're going to hose these things out of here. So I'm going to crank up the power on this, see if I can get this to an angle where you can see it, and we're just going to hose this down. I'm going to try not to introduce bubbles to it, but I'm trying to collect all of the cells that are floating in trypsin just by spraying the surface of the flask and trying to get every bit of it that I can. It's probably good for that one. So we'll take all that out and go for the other one. So I'm working the pipette around here to get all the cells off that I can. Just pipetting out and pipetting in. And once we have all of our cells out of here, we're going to pop them in the centrifuge and spin them down. And then we'll end up with a pellet of cells, a bunch of media and trypsin on top that we can suck up. Alright. That should do it. Take that out. Put it in a new conical. Turn the speed down. There we go. And now we have all of our cells floating around in there. You can't see them, but they're in there. So I'm going to take these out of the hood, put them in the centrifuge, spin them down, and then we'll have all the cells that we need. Sweet. Takes about 5 minutes. We won't make you watch all of that. Alright, so we got our cells all spun down here. This is just a balance, it's just water, we don't care about that. But with this we want to be a little careful because it's a little hard to see, but we have a little cell pellet right there at the bottom, and the rest of this is just media. So we're going to take that media off, put the cell pellet in our Bio-Ink, and then print. Got it. We got our cells all spun down, we have the right number. It's about 2 million for this experiment. And we're going to combine that with our polymer components to make Bio-Ink. And the long-awaited moment of 3D printing will begin. So I've got my cells in here. I'm going to rinse those out and put them in the hydrogel by using my Crosslinker solution. grab that. We want. It's pretty important to get all this out. I kept talking about how carefully formulated the recipe is. So I really want to get absolutely all of this. And we'll use this to grab the cells, gently pipetting to break up the cell pellet. The cells were all compacted because they just came out of the centrifuge. So this is spreading them out. And it's a little bit hard to tell, but this solution that I was working with was clear, and now it's very cloudy because it's full of cells. And I'll take that and add it into my solution of PEG alpha methacrylate and PEO, or polyethylene oxide. That's that shear thinning chemical that I told you about earlier. So I can see there's a cloud in here, and the cloud is the cells that I'm pipetting up and down. And I'm trying to very gently disperse the cells all through the solution. And they'll stay where they are because there's PEO in here that prevents them from settling or floating or dropping. But I can still pipette it up and down, so I'll be able to print with it. And there we have it. That is Bio-Ink. That is a hydrogel polymer precursor solution with cells inside. And now the fun part, we're going to load it up. We're going to start printing. So what I've done is I've already dismantled the build head of the printer so I can load all this up, but I'll show you how that works. First thing I'm going to do though is put my printing tray on here, go around from the back. Put that on the build head and then use our science rubber band to secure it. Yeah, here we go. This thing would slide around if I don't do this, so sometimes you need a special tool, sometimes you need a rubber band. So we got that on there. Like so. And I'll take, this is a syringe with a big blunt needle on it. And I'm going to use this big blunt needle to suck up all my Bio-Ink. Now I've got my Bio-Ink. I'm going to get the rest of the air out of that blunt needle. We'll take that off for disposal. And then we're going to put on a very fine needle to do the actual printing. The tip of this needle is 150 microns across, which means that when it starts printing, that's how thick the print dimension is going to be. So I'm purging the air out of this very, very thin needle. I don't want any bubbles in this print. Try and get all the air out of there. This is working pretty well. All right, we got a little bit of material coming out of the end. That means the needle's all loaded up. So we flip this around and then we put it into a couple of custom printed 3D parts here. And we'll attach everything. So this is called the build head. It's going to contain the syringe that has our Bio-Ink, and it needs to be screwed together. That's all going to go on there. And then I need to attach this screw, the clutch, so that when I put this on the 3D printer, there's a gear up here that is going to mesh with another part on the motor. And that motor is going to spin, and so this is going to spin, and that's going to push liquid out of the bottom of the syringe. And that's how it works, basically. So we load all this up, tighten everything together, grab a couple more screws, and now I'm going to take this apparatus with our syringe with the Bio-Ink and the needle, and I'm going to put it on the motor here. So this just meshes, screws in, pretty good. We don't want anything too tight because the plastic can crack, we want to be careful about that. And now we're ready to start printing. So I'm printing tubes, not super complicated. They're also not this big, this is zoomed in. but I can use this program on my computer to interact with the 3D printer. So if I want to move it in the Y direction, I can move it in the Y direction. If I want to move it in the X direction, I can move it in the X direction. I've got my 12-well plate there with all my life support, and I'm going to be printing inside of that life support. So I'm going to navigate this over to my first well, and then I'm going to drop it in, do all of this by line of sight, and line it up with my life support, drop it in more. It's like landing a spaceship. So less disastrous if it fails. And we'll lower it in. And this program is already loaded up, so it knows exactly what to print. There's something called G-code, which is a series of programming commands that the computer sends to the printer that translates into movement of the build head there. So if I want to make something like a tube, what I do is I model it on a solid modeling program like SolidWorks or Fusion 360. Then I put it into something called a slicer, which has settings for this printer and this material. There's a lot of work that went into that. That's all pretty much done now. And that generates something called G-code, which then goes from the computer to the printer. And if I hit print, we should see that this just starts going. There we go. So it's making a tube, spinning around. It's going to start at the bottom and work its way up. We'll see that on here, but this is how it goes. This is 3D bioprinting. Thanks for following me through that whole 3D bioprinting process. It was fun to get to show it to all of you. For anyone who's watching here, we got to do pretty much every single part of it, from prepping and weighing polymers to preparing the media solutions to dissolving everything today, getting the cells ready, getting the bio-ink ready, getting the printer ready, and then finally we got to see what the bioprinting actually looks like. Hopefully we'll get to see what the final prints look like too. We'll have to stay tuned for that because I won't know until tomorrow if they actually worked out. But thank you all for coming along on this with me. I hope it's been informative. I hope it's been helpful. And again, my name is Duncan Davis Hall, and we're in the Magan Lab at CU Denver Bioengineering out on the Entrance Medical Campus. So thanks, everyone.